In this video, we are going to learn about the digestion and absorption. Digestion means that the ingested food it is broken down into its simplest form because that is the form which is absorbed by the body. Digestion it is a catabolic process and intrinsic and accessory enzymes they are used to break down the macromolecules, and these enzymes they use the process of hydrolysis to break down the complex foods. And hydrolysis it means that water is added to break down the chemical bonds. Absorption is the process of moving substances from lumen of the gut into the body. We know there are tight junctions between the epithelial cells which are lining the gut. So the molecules they cannot pass between the cells. They have to pass through the cell to go to the interstitial space, and then they're taken up in the capillaries. Lipid molecules they have the advantage they can be absorbed passively through the membrane. We know that because our membrane is lipid bilayer, but other polar molecules they are absorbed by active transport. And most nutrients are absorbed before chyme reaches the ileum. Ileum is part of the small intestine. This is a small intestine cell. These are we know in the small intestine villi are present on the luminal side, on the apical side of the cell, which is the luminal side, and these villi they increase the surface area for absorption. So this is a cell with its microvilli, and the nutrient they pass through here where the blue arrow you can see. The first they cross the apical surface, and then they cross the basolateral surface, and then they go to the capillary blood. Now let's see how the nutrients are processed. We have 5 macromolecules. We will discuss digestion of each one of them. Let's start with the carbohydrates. Carbohydrates are their monomers are known as monosaccharides, and only monosaccharides can be absorbed. Starches and disaccharides they're broken down to oligosaccharides and then disaccharides, and the process it begins in the mouth. In the mouth we have salivary amylase. We know saliva it is secreted by the salivary glands which are present in the pharynx, and those glands they contain salivary amylase. Amylase it breaks down the carbohydrates into simple forms. It breaks down into lactose, maltose, sucrose, and then final breakdown it happens when these are broken down further into monomers. Now the process has already started in the mouth, and after mouth, when the food it reaches the stomach, in the stomach the salivary amylase it does not act because the pH of the stomach is very low because of the hydrochloric acid, and so amylase cannot work in such low pH. And then when the food it enters the small intestine, over there the pH of the food it again becomes alkaline because of the addition of the pancreatic juices. And in the pancreas there is also an enzyme known as pancreatic amylase. So amylase is for carbohydrates. Pancreatic amylase it breaks down the starch to starch or glycogen that has escaped the salivary amylase into oligosaccharides and disaccharides. And then after along with pancreatic amylase, we also have brush border enzymes. Brush border enzymes, they are dextrinase, lactase, glucoamylase, maltase, sucrase. They further break down these oligosaccharides and into lactase, maltose, sucrose, and then into monosaccharides, glucose, fructose, galactose. Monosaccharides, we know that this lactose it is broken down by lactase, maltose is broken down by maltase, and so these are the enzymes which are present in the brush border, and they act on disaccharides and they convert them into monosaccharides. Then monosaccharides they're co-transported across the apical membrane. of the absorptive epithelium, and they use a secondary active transport with sodium. So they are co-transported along with the sodium into the apical cell, and the energy it comes from the sodium potassium pump, which is acting at the basolateral surface of the cell. So that is why it is called secondary active transport, because that sodium potassium pump which is present at the basolateral surface of the cell, it is creating that concentration gradient for sodium, and because of that this secondary transport of sodium and monosaccharides it is happening. And then monosaccharides they exit across the basolateral membrane by facilitated diffusion. This is a chart. It summarizes everything which we have said. So the digestion for the carbohydrates, it begins in the mouth with the salivary amylase. Then the pancreatic amylase, it comes in, and the carbohydrates, the starches, and they are broken down into oligosaccharides and disaccharides. Those disaccharides are further acted upon by the brush border enzymes, and they are converted into monosaccharides. So these lactose, maltose, sucrose, they are disaccharides. They are further acted upon by brush border enzymes and converted into monosaccharides. And then these monosaccharides, they are absorbed from the apical surface of the cell. They go in the lumen of the cell, and secondary active transport is used along with the intake of the sodium. And then they, from the brush border, they go via facilitated diffusion. Now this will describe how it happens. Let's go over it step by step. So in the first step, this is the pancreatic amylase, and it is breaking down the starches, and it is going to break down them to oligosaccharides, disaccharides, and then the brush border enzymes are going to act upon them and turn them into monosaccharides. So these monosaccharides, then they are at the brush border, and now they are monosaccharides. We have brush border amylases, just like, for example, lactase, which breaks down lactose. I'm again and again saying lactose because we are very familiar with this lactose intolerance. So when this enzyme is absent, then lactose intolerance happens. So these monosaccharides, they reach the apical surface of the cell, and then they are taken into the lumen of the cell along with the sodium. This is known as co-transportation, and the energy it is coming from this pump. This is secondary active transport. Energy is coming from this pump, the sodium potassium ATPase, which is present on the basolateral surface. So these monosaccharides, then they come in and then they diffuse to the basolateral side, and from the basolateral side they go into the interstitial space and into the blood, and to leave the basolateral side they use facilitated transportation. Now, what can go wrong? Just like I said, that there can be lactose intolerance when certain people, they are deficient in lactase. They cannot consume lactose. So any lactose eaten, it remains undigested, and it creates an osmotic gradient. So when lactose is present in the lumen and it cannot be absorbed, the water it comes into the lumen, and this results in diarrhea. And it can also pull water from the interstitial space into the intestinal lumen. And then because there is undigested solute present in the intestine, then bacteria act on that, and they get bloating, flatulence, and cramping pain. Treatment is to add lactase enzyme drops to the milk, or take lactase tablet before consuming milk products. Now this brings us to the protein. So the digestion of the protein. First of all, let's see, the source of the protein is not only dietary, it also includes the digestive enzymes which are present in the body, and proteins from the breakdown of the mucosal cells. So we have three sources of protein: dietary protein, that the digestive enzymes, and the proteins from the breakdown of the mucosal cells. And proteins, they are broken down into large polypeptides, small polypeptides, and small peptides, and into amino acid monomers. So these are the different digestive compounds of protein which are present. Now, digestion of the protein, it begins in the stomach, and there is a substance known as pepsinogen which is present in the stomach. Pepsinogen is the inactive form because it is not present in the active form. Otherwise, it can digest the stomach itself. So, pepsinogen, it is converted to pepsin at the pH of 1.5 to 2.5, which is a very low pH which is present in the stomach, and it becomes active in the high pH of the duodenum. In the small intestine, the pH, it is alkaline, it is not this acidic. So this pepsin, it becomes inactive in the duodenum. Now we are in the stomach, and the digestion of the protein has started with the pepsin. Now let's quickly go over this, and then we will read the details. So this is protein, and the digestion is starting in the stomach. This protein is broken down into large polypeptides. Then in the pancreas, in the small intestine, there are pancreatic enzymes, trypsin and chymotrypsin, and then carboxypeptidases. They also act on these polypeptide chains and small peptides, and they further break them down. Then in the brush border, we have amino peptidase and carboxypeptidase, which remove substances from the amino and the carboxy end of the peptide, and they make them into more smaller compounds and amino acids. And these amino acids, they are then taken up at the apical surface as a co-transport, and then they are from the basolateral surface, they enter the blood. Now, this is just what I said written in this form. So now we already, just as a reminder, the digestion has started in the stomach with the pepsin. Where did pepsin come from? Pepsinogen was converted to pepsin. And now we are in the intestine. In the intestine, the pepsin, it has become inactive. And now in the intestine, we have pancreatic proteases. Pancreatic duct, pancreas is a gland in the pancreatic duct. It opens in the small intestine. All right, along with that for the bile. So pancreatic proteases are trypsin and chymotrypsin. They cleave the protein into smaller peptides, while carboxypeptidase takes off one amino acid at a time from end. And then we have brush border enzymes, which are aminopeptidase and carboxypeptidase and dipeptidases. They break down oligopeptidase and dipeptides into amino acids. Now, amino acids, they are co-transported across the apical membrane, they are co-transported via sodium or hydrogen. So along with the amino acid, the sodium or the hydrogen, it goes in, and this is secondary active transport. It means that the energy, it is coming from the sodium potassium pump, which is acting at the basolateral surface. And then the amino acid, they exit the basolateral surface via facilitated diffusion. So this is almost the same. Their apical part and the basolateral part, it is the same as the carbohydrates. Now let's look into this diagram. Let's go over it step by step. So this is the pancreatic peptidases and proteases. They break down uh, the complex polypeptides into amino acids or dipeptides, and those dipeptides are further broken down into amino acids by the brush border enzymes. Now let's see. So this was now the second. Then the brush border enzymes, they come into play, and they are breaking down the dipeptidases into amino acids. And then these amino acids, they are taken in as a co-transport, and this is a secondary active transport. The energy is coming from this pump. This pump is making the ATPase, and it is keeping the sodium gradient low inside. And this low sodium gradient, this helps this sodium to come in, and the amino acid, it is co-transported along with it. And now, through facilitated diffusion, it comes out of the basolateral surface and it goes into the blood. So this is our protein digestion. Now let's see what happens to the lipid. So lipid digestion is somewhat different. So you will have to remember this carbohydrate and protein, they are the same, and only the enzymes are different. For the lipid, it is different. First of all, we have a lipase. The enzyme which is going to act on the lipid is known as lipase. We have lipase in some amount in present in the mouth and in the stomach, but they are insignificant. So basically, the digestion, it starts in the intestines, in the small intestine. First of all, what happens? There is emulsification. What is emulsification? Maybe you're familiar with this term. When there is a greasy pot or something, we put detergent on it, and that detergent, it emulsifies the fat, right? It breaks down the fat. So what is emulsification? Triglycerides and their breakdown products are insoluble in water. They need pre-treatment. Just like, we put some soap in a very greasy dish and leave it for a while so that it is easier to clean. So similarly, the triglyceride and their breakdown products, they also need pre-treatment because they are insoluble in water. And the pre-treatment, the soap for these triglycerides and these fatty substances is bile salt. So bile salt, when the fat comes in the intestine, just imagine that the bile salts are sprinkled on them. And these bile salts, they break the large fat globules into smaller ones. And then the digestion starts. The pancreatic lipases, they break down the fat into monoglyceride plus two free fatty acids. So now, because before that the fat, it was one big molecule, and the digestive pancreatic lipases, they were unable to act on it. So first of all, the body coated them with the bile salts. The bile salts, they were applied to that big fat molecule, and that big fat molecule, it is broken down into smaller fat globules. And then the pancreatic lipase, it breaks down fat into monoglycerides plus fatty acids. And then now these fatty acids, we know the fats, they cannot travel in water. So now the body has to find a way to make them travel into water. What does it do? So this is all happening in the lumen. Okay, this is all happening in the lumen. And then micelle is formed. What is micelle? Products from digestion become coated with bile salts and lecithin. Now these micelles, they are smaller than this emulsification step. So now the lipases, they have broken down the big fat globule into a simpler form, and micelles, and those simpler forms are again coated with bile and lecithin, and they form small particles known as micelles. Those micelles, they carry that fat to the brush border. And then in diffusion, the lipid products, they leave the micelle. So micelles are just like, they're what can I say, a vehicle, a cab, right? So the fat molecules, they are like sitting in the micelle, and now they go to the brush border of the intestine. And then they get out from the cap. So they leave the micelle and they cross the epithelial membrane via diffusion, because now is not the problem. Before that, they were in the aqueous environment, but now they have reached the cell membrane, and we know the cell membrane is lipid bilayer. So lipids, now they can simply diffuse inside the intestine. So this is important to remember, that these micelles, they are present in the lumen, and they carry the small monoglycerides and fatty acids. They carry these simple form of lipids to the brush border, and these micelles, they are formed by coating these simple forms with the bile and the lecithin. Now let's see what happens inside the cell to these, because the cell, it is again going to convert them into a complicated compound. Why is this not letting me? All right. So now we are still in the intestine, but now that those lipid, it has diffused inside the cell. Now it is in the cell of the small intestine. Over there now, chylomicron is formed. That lipid, that is packed into chylomicron. What is chylomicron? Lipid products are converted back into triglycerides and packaged with lecithin and lipoproteins. So they are converted back to triglycerides and they are packaged with a substance known as lecithin, and they're coated with proteins, lipoproteins, and this thing is known as chylomicron. So micelle was present in the lumen of the intestine when the fat was absorbed. It was just like a vehicle on which the simple form of lipid it sat, and it was taken to the it was shuttled to the apical surface, you can say. And now this chylomicron, it is being formed inside the intestinal cell. It is inside the cell, and it contains lecithin and lipoprotein. And now these chylomicrons, they are transported by exocytosis. So they by exocytosis they move out from the basolateral surface, and now they are in the interstitial fluid. And when we were doing the lymphatic system, I mentioned it over there too, that one of the job of the lymphatic system of the lymphatic vessels is that they absorb the chylomicrons. Now these chylomicrons, they are present in the interstitial space, but they are too big to enter the capillaries, to enter the capillary. So they enter the lymphatic vessels. And those lymphatic vessels are known as lymphatic lacteal. They are very whitish, they make very whitish fluid, and they enter the lymphatic lacteals. And eventually they are emptied into the venous blood at the thoracic duct. So those carry them to the circulation. So this is how fats are absorbed. It is interesting. It is a little different. So then once now the fats are in the blood, we are still not done. So what happens? The chylomicrons are broken down into fatty acids and glycerol by lipoprotein lipase. Okay, so in the blood they are broken down by lipoprotein lipase into fatty acid and glycerol, so cells can use these. And short chain fatty acids can diffuse directly into the blood. So this is about lipids. Now let's move on to the next slide. All right, so these are all the steps we discussed about that. So here there is in the mouth, like I said, there is lipase present, but it is insignificant. Similarly, in the stomach too. And then finally, when we come to the small intestine, the bile it comes from the liver, and it enters the small intestine. Bile it is stored in the gallbladder and it comes in the small intestine, and then it acts upon the big fat molecule. It emulsif First of all, the emulsification happen. The big fat molecules, they are broken down into smaller fat particles, and then the smaller globules, they are acted upon by the lipase present in the pancreas. The pancreatic lipase, it breaks them down to their simple forms, to their monomers and fatty acids, to glycerol and fatty acids. And then those simple forms, they are made, they ride on the micelle. So micelle is that again they are coated with the bile salt and lecithin, and they are carried to the brush border. And in the brush border they get off the micelle and they diffuse into the cell. In the cell they are changed into chylomicrons. Again they are made into triglycerides, and then lecithin is added and lipoprotein is added on the outside. And those chylomicrons, they are removed from the cell, from the basolateral surface, by exocytosis, and they are taken by the lymphatic lacteals. Lymphatic lacteals take them to the circulation, to the blood circulation through thoracic duct on the right side, remember. And then in the blood there is an enzyme known as lipoprotein lipase. It breaks them down into glycerol and fatty acids so they can be used. Those chylomicrons are broken down by the lipoprotein lipase into glycerol and fatty acids so they can be used by the cells. So this is about the fat. It is a little different, but it is very interesting. All right, so this is again that in the visual form. Let's see, let's go over it again. So this is a big fat globule, right? Triglyceride. It has come in the intestinal lumen, and now the bile salts attached to this thing, and they break them into smaller globules. Now these globules, it is easy for the lipase, the pancreatic lipase. It acts on these things, and it converts them into their simpler forms. Those simpler forms are again attached to the bile salt and lecithin, and this thing is now known as micelle. So these micelles, they carry them to the intestinal brush border, and in the intestinal brush border they get off from the micelle, and they diffuse in the lipid bilayer, and they go inside the cell. Now inside the cell they are made into chylomicrons. Chylomicrons contain they're again made changed into triglycerides, and then they're packed in chylomicrons. They're packed in packages. Those chylomicrons, they contain triglycerides, and they also contain cholesterol. I forgot it. Cholesterol is also packaged inside, and because they are all hydrophobic, so they are kept inside, and on the outside we have a lipoprotein which is polar. So then these, because they have to go in the aqueous environment, remember, even the interstitial fluid, it is aqueous. So the lipoprotein is on the outside. On the inside we have triglycerides, we have cholesterol, and then they diffuse through the basolateral surface. These chylomicrons, they diffuse through the basolateral surface. They go in the interstitial fluid. Interstitial fluid, there are lacteals, right? So they have diffused here, and when they came out of the cell, they were chylomicrons, and they are lacteals. Lacteals, they are lymphatic vessels because they cannot enter the capillary because they're big molecules. These lacteals, they carry them to the circulation through the thoracic duct. All right, and now our last macromolecule, nucleic acid. Let's see how nucleic acids, they are digested. And so nuclei of the ingested cells in blood, they contain DNA and RNA. These are our nucleic acids. And the pancreas, just remember pancreas. And for the lipid, bile also comes in. Okay, so the pancreatic nucleases, they hydrolyze the nucleic acid into nucleotide monomers. Then at the brush border, there are enzymes, nucleosidases and phosphatases. They break nucleotides down into nitrogenous base, pentose sugar, and phosphate ions. And these breakdown products are actively transported by special carriers in the epithelium of the villi. So they have special carriers on the apical surface. Now this is about the absorption of these nucleic acids. Again, the pancreatic enzyme, they start the process, and then the brush border enzyme, they turn them into simpler form, and those are absorbed from the apical surface through special transporters. This was about our macromolecules. Now let's see about the absorption of vitamins. So electrolytes and water. So first of all, let's go over vitamins quickly. We know we have two types of vitamins. We have fat soluble vitamins, and then we have water soluble vitamins. So for the fat soluble vitamins, they are A, D, E, and K. They are carried by the micelles. Remember, in the intestinal lumen there are micelles, and then just as a revision, I'm repeating this. The fat in the intestinal lumen, they are made into micelles, and then when they diffuse into the intestinal cell, inside the intestinal cell, they're packaged as chylomicrons. So they leave the basolateral surface exocytosis as chylomicrons. So these fat soluble vitamins, they also take a ride along with the fat because they're fat soluble, so they're present in the fat. So that is why for the absorption of the fat soluble molecules, we need a little bit of dietary fat. We need to take in a little bit of dietary fat. If someone is taking these vitamins as a pill, but there is no fat in their diet, they will not be absorbed. So fat soluble molecules, they are carried by micelles, and they diffuse into the absorptive cells. So because they are inside the fat and fat can cross the plasma lipid membrane, the fat hops off the micelle and it goes inside the cell. And then along with that, they follow the same path along with the fat and they are absorbed. And the water soluble vitamins, especially the C and the B, they are absorbed by diffusion or by passive or active transporters. Now the vitamin B12, it has a special protocol. So vitamin B12, it is a large charged molecule in the stomach. It attaches to intrinsic factor. So the stomach cells, they secrete a factor known as intrinsic factor, and it attaches to intrinsic factor. It is important for vitamin B12 to attach with the intrinsic factor. And then this intrinsic factor, vitamin B complex, it goes into the small intestine, and in the small intestine, it is absorbed by endocytosis. If vitamin B12 is not attached to intrinsic factor, it is not absorbed, and it results in anemias, because vitamin B12, it is, if you remember, it is required for the rapidly dividing cells, because it is important in DNA division. So we need vitamin B12 in rapidly dividing cells, and red blood cells, they're rapidly dividing. So many times when vitamin B12 is absent, megalocytes, they develop because the cell cannot divide. All right, and then in large intestine, there is vitamin K and B, vitamin B from bacterial metabolism. They are absorbed. So our bacteria also, they because of their metabolism, we get some vitamins and they are absorbed in the large intestine. So this was all about the absorption of the vitamins. Now let's go towards the electrolytes. So absorption of the electrolytes, most of the ions are transported actively along the length of the small intestine. Okay, so there is active transportation all along the length of the small intestine. Iron and calcium are exceptional. Iron and calcium, they are absorbed in the duodenum. Duodenum is the first part of the small intestine. Sodium absorption is coupled with active absorption of glucose and amino acids, like we saw, right? And chloride is transported actively. Potassium diffuses in response to osmotic gradients, but lost if water absorption is poor. And usually amount in the intestine is the amount absorbed. So the most of the electrolytes, they are absorbed in this way. Now let's go ahead and let's see what is different about iron and calcium. I'm sorry, it's taking a little while for me to change the slide. All right, so the iron and calcium, their absorption, it is related to the need. Okay, so the rest of the electrolytes, they are being absorbed freely along the length of the small intestine, but iron and calcium, they need to be regulated. Their absorption needs to be regulated. Um, Iron is stored in the mucosal cell with the ferritin, because we know iron, it cannot, it makes compounds in its reduced form. It can be dangerous. So iron, it is stored in the mucosal cell with the ferritin, attached to the ferritin, and it is transported in the blood attached to transferrin. There is a protein for the iron which is known as transferrin. So and iron, its absorption, it is regulated by the amounts of these presence of these different proteins. And if the body already has too much stores of iron, then less iron is absorbed from the intestine. So it is regulated. Similarly, calcium absorption is regulated by vitamin D and parathyroid hormone. Remember, parathyroid hormone is the one which absorbs the calcium from the intestines, from the bones, from the kidneys, from everywhere, because its job is to maintain calcium level in the blood. So calcium absorption, it is regulated by vitamin D and parathyroid hormone. Now let's see about the water. So there is 9 liters of water present in the GIT tract, mostly from the secretions. It enters the small intestine. 95% of this is absorbed in the small intestine by osmosis. Most of the rest is absorbed in the large intestine. And so this is about water. Now what can go wrong in this absorption and digestion process? What can go wrong is, I just want this to go. All right, so there can be malabsorption, and malabsorption, it can be caused by anything that interferes with the delivery of bile or the pancreatic juice. So anything which hinders the delivery of bile and the pancreatic juice, it is going to cause malabsorption. And damaged intestinal mucosa from bacterial infection or some antibiotics, they can also cause malabsorption. And then there is gluten sensitive enteropathy, which is known as celiac disease, and it is a common malabsorption disease. In this, there is immune reaction to the gluten, and that immune reaction to the gluten, it causes damage to the cells of the intestine, and intestinal villi and brush border, they are affected in the celiac disease. And what is the treatment is that gluten, it is eliminated from the diet. So this is all about absorption and digestion.